All right, everyone. For today, we have a topic called complement system. Let me write it down. Complement system. A very important topic of the immunology. Now, complement is nothing but a heat labile protein or factor which is present in plasma. Okay, so that you understand where complement is present. Next is, it's also known as a factor, and the role of this is basically enhance the killing of microbes. So as you can imagine right over here, microbe is there, an antibody is there, and complement is going to enhance this process of antibody mediating killing of the bacteria. So it's going to enhance the antimicrobial activity of antibiotics. Okay, and there are how many proteins? 30 proteins, serum proteins, that are going to come under this entire complement system. If we talk about functions, there are various functions that are involved with complement pathway. One is phagocytosis. So just imagine this is a phagocyte, and inside that you can actually see it's trying to eat that particular cell. So phagocytosis. You have microbes, and microbes, they can target they can target with complement and also with antibodies, right? So that's another function of this. Next is movement of cells, that is chemotaxis in one particular direction. So chemotaxis is very, very important feature that is there because of the complement. Next is lysis of bacterial cells. So as you can see, I've created that dotted line. Dotted line is nothing but showing lysis of the bacteria. Lysis of the bacteria. Next, you have complement is nothing but it's a bridge between innate and adaptive system. Very, very important point. The bridge between the innate immunity and the adaptive immunity is the complement pathways. What it does is basically enhance the antibody response, right? So second one is memory of the immune system is also associated with complement. Next one is there is a lot of waste that is there after killing of the microbes. Maybe the immune complex is there. So these are antibodies. You have antigens, and this is an immune complex, antibody-antigen complex. That is also the thing that should be removed. You have inflammatory mediators because of the inflammation. And then you have dead cells, right? Host cells, they are dead, so they need to be taken care of. So all these things are being associated with complement. Okay, now let's talk about complement activation pathways in general. Let's understand the introductory features of complement activation pathways. So there are three pathways. The first one is alternative pathway. That's the first pathway. Second one is lectin pathway. Easy to remember, right? So third one is classical pathway. And what are the common features that are associated with complement pathway? You have, let's talk about one by one. So the first important feature is you have a protein that we have already understood that we have a protein in our plasma, and that protein is present in inactive form. Okay, so the next thing that is going to happen is conversion of this to active form, and that will happen because of the enzymatic degradation. So enzyme basically is going to degrade this particular inactive protein into chemically active protein, and that is going to further initiate cascade of the activation process. And as you can see, this chain reaction is going to activate other proteins, and then you'll have a chain of activation, the chain of events that's going to happen. And further final outcome of that, all these pathways, bacteria is going to get a lot of these complement pathways, proteins that are going to cause opsonisation. Next is inflammatory mediator production is going to happen. So let me write it down. Inflammatory mediator production is going to happen. And the final one, most important feature, is the complex formation, which is called MAC, membrane attack complex. That is going to kill the microbes. Very, very important complex. Okay. Now let's move to the first pathway, and let's understand the first pathway in detail, which is alternative pathway. Very, very important topic. First one is how it get activated. Bacterial molecules, specific molecules that are present in bacteria, the molecules that are containing repetitive structures like this. Okay, repetitive structures, and the example, one of the important example is LPS, which is lipopolysaccharide. Is a molecule composed of lipid and also composed of sugar moiety. this can activate this pathway. What happens is you have C3 protein that is going to, converted into two different proteins, which is C3a and then C3b. Okay, so degradation is going to happen. And then what this protein is going to do, specifically C3b protein is going to bind to the bacterial surface. So I'm just quoting bacterial surface with C3b. The next point is you'll have factor B. Factor B is going to bind to this C3b. Everything is happening step by step. So you have this pathway where you have factor B is attaching. Next one is factor D. Factor D is going to cleave the factor B. Okay, it's going to cleave the factor B. And then final outcome is we are going to have C3 convertase formation, which is C3b, capital B and small b. That's the C3 convertase enzyme. And this particular protein is going to basically, if there is another C3b that is going to attach to this particular complex, it becomes C5 convertase. So from C3 to C5 convertase formation is going to happen, right? So you already have C3, which is going to convert C3 into C3b. Here, C5 is going to convert C5a, C5 to C5a and C5b. Okay. And then C5b, what it's going to do is it's going to combine with various factors, factor 6, factor 7, factor 8, and 9 to form MAC, membrane attack complex. And that is going to cause pore formation, and that is going to cause cell lysis. So this is the overview of your alternative pathway. Very, very important pathway. Next, you have second important pathway is, before pathway, let's understand the inflammatory effect of the proteins, because that was associated with membrane complex and opsonization. You have another protein, which is C3a and C5a. These two proteins, they are inflammatory mediators. They are involved in inflammation, right? So the alternative pathway, you have seen that you have those proteins forming membrane complex. The other proteins, they're going to cause vasodilation, C3a and C5a. You have vascular permeability. That is going to happen because of this. You have nerve stimulation. Next, you have the phagocytes that are going to get activated by these particular proteins. And specifically, if I talk about C5a, which is chemotactic factors. Chemotactic factor. And this is chemotactic factor for a specific cell. I'm drawing this cell, okay? I don't know if you can remember this. I've discussed this point in the previous lectures also. This is neutrophil. Let me write it down. Neutrophil is going to get attracted with C5a, very, very important proteins. What it's going to do is it's going to, target those microbes. Phagocytosis is going to happen. Look at that. Microbes are getting inside, and then ingestion of these microbes is going to happen, and destruction of the microbes is going to happen. So phagocytosis is going to get activated on its full scale. Now let's talk about another pathway, which is lectin complement pathway. Okay, lectin complement pathway. How that happens? You have, mannose-binding protein, MBP, and that is there in our body, and it is being produced by liver. So this is the schematic for liver, and liver is going to produce it as acute phase protein. Acute phase protein, you have a mannose-binding protein. As this is simple word, that means it's going to bind to bacterial mannose, right? So first activity that it's going to do is it's going to bind to the mannose residues that are present on the bacterial surface. So just imagine this is bacteria, right? And these are mannose, and it's going to bind over there. So it basically is going to act like opsonin. Next is it is also able to bind to viruses on the envelope of the viruses, because you also have residues over there. So virus is going to get coated with this, bacteria is going to get coated with this, and also your antigen antibody complex is going to get coated with this. Okay, so it basically is going to work on all these three important components, and then the function is basically, I told you, first one, opsonin. So it will enhance the phagocytosis because it's attaching to the bacteria and viruses. It's going to activate MASP, which is mannose-associated serine elastrase. Serine elastrase. Let me elastrase. Okay, elastrase. So that enzyme, and it's also very, very important for C3 convertase, right? And if you have C3 convertase, then you can imagine how the activation of the alternative pathway is going to happen, right? Okay, now let's move to the classical complement pathway. very, very important pathway, which is classical complement pathway. This pathway is initiated by antigen antibody complex. So I'm drawing antigen antibody complex. So you can see these Y-shaped structure, they are antibodies. And then you have antigen, and they are binding and forming this complex. So this pathway is going to initiated by antigen antibody complex, right? And then along with that, it can also be initiated by microbial products. There are several products that can actually start this pathway. So what happens in this particular pathway is when you have antigen, let me label it antigen. Let me draw first the antigen. This is antigen, this is antibody. And then this is a specific protein that is C1 complex. It's going to bind to this particular complex, antigen antibody complex, by C1q. So this is C1 complex, and it's going to bind to antigen antibody by C1q. Okay, and then further, it's going to cleave C1. Okay, this is going to get cleaved. C1 is going to get cleaved to C2 and C4. This is the classical complement pathway, C2 and C4. You have these proteins that are getting produced, and then you have C3 convertase, which is, in this case, again, you have C3 convertase. In this case, you have C4b and 2a. These proteins are going to convert C3 to C3a and C3b. And you know that after this, you will have complement cascades that are going to get activated. You are going to have opsonization. So opsonins, they are going to again get activated. You will have inflammatory mediators that we have already talked about, right? C3a, C5a, and then you have a membrane complex formation, and that is going to kill the microbes in a very, very quick way. Okay, so in this, I've tried to cover all the important pathways, and I've also tried to cover the features of complement. I know it's a little faster, but on the course website on Research Goal, you have the availability of notes, so you can check the website and log into the course. You will get a lot of different content, right? On YouTube, you will have the videos available, but the content associated and the certification, if you want to complete, is there on Research Goal. And Research Goal, everything is more organized. Right over here, you will have videos scattered everywhere. So the courses are more organized are there. So if you want to have a good organized learning experience, in what way you have to complete all those videos to get information on Innate Immunity, please check it out. Link is there in the description box, and let me know what was your experience in overall completing that particular course. I'm looking forward to meet you over there. All right, thank you so much. Let's meet in the next video.